other speakers who set up my study in a really nice context of human-induced selection and invasion, things that I'm going to be talking about. Hawaii is one of the most isolated archipelagos on Earth. It's located about 3,000 miles from the nearest. Do you have that?
Yes.
Okay, I'll just stop wondering. So it's located 3,000 miles from the nearest landmass, and as a result, in part of this isolation, as well as other things, Hawaii is a real hotspot for adaptive radiations. And so we know things like land snails, Hawaiian silver swords, and Hawaiian honey creepers, which are a group of birds that are all derived from a single ancestral finch species, and they radiate into about 55 species. This is about half of them. Unfortunately, in addition to being a really hotspot of adaptive radiations, Hawaii is also an example of repeated biological invasions as a result of human introductions. And so there are many examples of human-introduced species, but the most recent one that I'll be talking about today is the combined introduction of house mosquitoes, which were introduced to Hawaii in the 1800s, and then Plasmodium relictum, which is the parasite causing avian malaria, and it was introduced in the 1940s. So since its introduction, at least 6 honey creeper species have gone extinct, and of those that survive, all of them are completely restricted to these high elevation mountaintops where the mosquitoes cannot live because it's too cold, and so there's no malaria in these mountaintops. Recently, however, in the past couple of decades or so, people have started noticing the increasing abundance of the Hawaii amakihi, shown here, in these low elevation habitats where there are tons of mosquitoes. And you can catch amakihi and find that they're really heavily infected with Plasmodium, and they seem to be doing great, and they have no fitness consequences that we can see. And so this is a really exciting example of adaptation that has occurred really rapidly. And so I have the same questions that many of us have and that people have already talked about in this session, although I'm not going to get to the punch of my question until the end. So I'm really interested in the system for how, what are the genes that actually underlie adaptation to malaria? Did tolerance evolve once and then spread among islands, or did they evolve independently on multiple islands? And then are there sort of genomic signatures that we can see in the stages of adaptation to malaria? So I'm mostly going to be touching on this first question for this talk. And so I also used genomic and transcriptomic tools and bioinformatics pipelines that I've developed, but I have to really highlight that this work was completely made possible by Taylor Calgary, who's a graduate student in my lab when I started, and she sequenced the amakihi genome and also developed an array of SNPs distributed across the genome. So she really allowed me to get a lot of this work done. So using these tools, we use a multi-pronged approach to studying adaptation. It's difficult in many cases to do experiments on vertebrates, but we have we like to use different approaches, as other people have mentioned, to really kind of strengthen the evidence for how adaptation occurs. And so in Hawaii, we have independent replicates on separate islands of evolution of malaria tolerance. And then within islands, we can have various elevational transects running from no malaria resistance at the top to high malaria tolerance at the low elevations. And so we can compare the genomes of these birds out in natural populations. We also have done a couple of experimental infections with malaria, and so we can compare the genomes of birds that survive and die from these infections. And then we have this amazing resource of just a tremendous amount of collection data. So we have tons of amakihi specimens that have been collected over the last 150 years or so. And so we can get we can compare genomes of birds before malaria introduction to contemporary birds, as well as kind of a time series throughout since the introduction of malaria to see which genes have been changing in frequency since then. And then the idea is to find genes that are going to be important in all of these different approaches, and those will be the strongest candidates for malaria tolerance. So I'll just talk first about this sort of spatial approach, and I'll just be focusing on samples that have been collected on the island of Hawaii, and none of these were collected by me. There's been tons before my time by a lot of really amazing scientists. And so I just subsampled 125 Amakihi from across the island at various elevations and in different populations. And so I just want to clarify that what I consider, the rest of the talk, what I consider low elevation are elevations that have mosquitoes and where the birds have experienced this recent selection pressure from malaria. And then high elevation are elevations that do not have mosquitoes or malaria transmission. So if we compare the genomes of birds at low and high elevations, they're mostly very similar, but there are a few outlier loci. They're really highly differentiated between elevations. And many of these have no sort of, I have no reason to believe they're actually involved in malaria. And that's not too surprising because there's a lot of other things that differ between low and high elevations that are exerting selection on the birds. But there are also several genes that do play a putative role in at least immune response, and these are a few of them. So there's this beta defensin, which we'll actually see later on in the talk to be important, and it's a gene that encodes for antimicrobial peptides. It's not really known if this is also helps resist malaria infection, so Plasmodium is not a microbe per se, but it's a pathogen. And so then there's also interferon genes that are known to play a role in pathogen defense. And then cell adhesion, which are sort of cell-cell communication, and then host-self other kind of recognition. And I put this one CD59 in here because I found it very interesting because it's related in function to a gene that's actually known to be important in human malaria resistance. Okay, so that what I just showed you was a result of pooling all low elevation into one sort of population, and then all high elevation individuals into one other population. But it seems plausible that each low elevation population might be evolving independently. There is some gene flow among populations, but they also, it's feasible that they could have different mechanisms for adaptation. So I repeated this with each population pair, so each low to high elevation pair, and did the same thing, and see, again, a lot of mostly similar genomes, but then there are a few outliers. And among those outliers, there's kind of a whole suite of genes that are involved in the immune system. And so this whole table are genes that are immune related, but the green ones are actually genes that are similar or that are found in multiple population pairs. And so they're common across the low elevations. And so again, we see the beta defensin coming out and interferons. We actually see some more genes that we didn't know about by pooling that pop out as important when we analyze population pairs separately. Okay, so the strongest candidates from this sort of spatial approach are beta defensin, as well as the other things that came out with population pairs, interferons, cells, adhesion, and major histocompatibility complex. And so the next step is to figure out if any of these genes are also important directly in conferring survivorship to malaria when the birds are infected experimentally. And so I used an experimental challenge done by Carter Atkinson back in 2000, and I used 2 survivors and 4 mortalities from this experiment. And I performed a genome-wide association test to see what are the SNPs that are actually most strongly associated with malaria survivorship. And so there are about 250 genes in that sort of upper tail there that are most strongly associated with survivorship. And so I took those 250 and classified them in terms of what their general function is. And some of the general functions are really baffling to me. They're not intuitively related to immune function, like olfaction. But then there are some large classes of gene function that do make intuitive sense for immune function, including things like heat shock protein that would be important, and inflammatory response genes. And one of these inflammatory response genes is actually beta defensin. So we can see from using these 2 approaches that we've sort of been able to hone in on one gene that has multiple lines of evidence for it playing a role in adaptation. And this next approach is pending funding from NSF, which I'm sure will happen any time now as soon as they realize how exciting it is. But I just want to mention that we have been able to get really good data from this is just a plot of another museum specimen using our same approach. And then some other things that I'm currently working on are expanding this data set to other islands, and so I've been sampling Kiki across different islands along similar elevation gradients, as well as a couple of other species in the honey creeper clade that are found on multiple islands. And so I'm using the same genomic approach as well as measuring gene expression at low elevation and high elevations. So this will allow me to get at that question that we all have of, is adaptation happening the same way across replicated species and populations? So we also have another experimental challenge that we just completed, and this is in collaboration with Carter Atkinson again at USGS and Christina Paxton at UH-ELO. And so we have more samples to look at here, so we'll have more statistical power, but then we also were sampling gene expression throughout the course of the infection to see what genes are invoked right at the time of infection versus when the birds clear the parasite or don't. So to kind of answer that original question, there are some really good, strong candidate genes that we have for conferring adaptation to malaria. And the next step will just be to answer this, whether those same genes are involved across the Hawaiian Islands archipelago and in other related species. So I guess you'll have to tune in for next year. So I have a bunch of people to thank, and I think before I open it up for questions, I would like to put in a plug for graduate students and the lab manager. I'll be starting up a lab at the University of South Florida this fall, and I'm looking for people to join me. So feel free to come talk to me afterwards and check out my website if you're interested. Just let me know.
Some questions?
So you identified a few genes that are associated with an immune response that you think might be important, but it's interesting because the birds aren't really resisting the parasite, right? They have really high circulating parasitemias. And so given that you say it's tolerance, do you have any idea how an immune response or how MHC could help the birds not die even when really heavily infected?
Yeah, that's a good question, because so they're living with high parasitemia and they're not actually getting rid of the parasite, they're doing something to deal with it. So you can imagine that maybe the immune response is sort of, I don't know, allowing them to enter the cells without doing damage or something like that, but they're clearly invoking some other things to allow them to survive. Yes?
So why only one species managed to evolve this resistance?
So there's actually some evidence that other species have evolved some resistance. The other two that I had pictures of at the beginning, and there have been experimental challenges done on those two, but the resistance or tolerance is just much lower, so there's not as much variation. But that's a really interesting question because they all have a common ancestor. You would imagine that maybe all 55 or all the remaining species would evolve resistance. And so it's a really interesting question as to why they don't. And I wish I knew. I would love to know.